Hello everyone, welcome back to today's lecture series. As a continuity of the lecture series, in today's video, we will be discussing about lactic acid bacteria and fermentation products. Let's dive in detail and let's learn this concept in a more simplified way. So today's topic for discussion will be about how the traditional techniques differ from the modern starter cultures or modern methods in preserving the food, and also a synthetic preservation and its effects. And we'll discuss about how basically the lactic acid bacteria are beneficial and how are its characteristics and how they are used in food preservation and a little bit more in detail. So what are basically lactic acid bacteria? So lactic acid bacteria are one among the beneficial microorganisms that produces lactic acid during fermentation, right? As we all know, because they are commonly known for the food preservation and flavor development for centuries and over these years. So they are basically the gram-positive bacteria, and they are acid tolerant, important in food preservation. If you take examples, yogurt, pickle, and cheese are the food products where lactic acid bacteria plays a major role. And over here, let's discuss about the characteristics or the microbial taxonomy of LAB. So basically, as I said before, they are gram-positive bacteria. So LAB survives in acidic conditions and prevents spoilage organism from growing. They are non-spore forming, anaerobic fermentation, and they produce lactic acid. So if we talk about the growth or the common area where they usually are present, or they require oxygen, nitrogen sources, vitamins and nucleic acids to grow. And their species include lactobacillus, lactococcus and streptococcus strains. So the biochemical pathway, if we take about the fermentation pathway, we have homofermentative and heterofermentative. So if you see homofermentative, they mainly produce the lactic acid. And over here, we have high acidic production. So what basically happens is, it processes sugar with the help of glycolysis pathway, where the aldolase is going to split the glucose into two pyruvates. As a result, they are directly converted into the lactic acid with an efficiency of 90% yield. So when you take the heterofermentative pathway, they produce lactic acid, carbon dioxide and ethanol. So what happens here is, they'll use the 6-phosphogluconate pathway, where glycolysis metabolism is going to happen, as a result of which we have the production of lactic acid, carbon dioxide and ethanol, which results in the carbonation and delicate sensory esters formation over here. So over next slide, we have about the dairy fermentation. So in case of dairy fermentation, we basically talk about the casein coagulation. So what happens here is, the LAB converts the lactose into lactic acid, causing the milk to coagulate and form the curd. So they're also used in yogurt fermentation, cheese production, and also in pH reduction. So the important starter cultures we use in case of dairy fermentation include Streptococcus, Thermophilus, Lactobacillus bulgaris, and major products are yogurt and cheese. So next we have about vegetable fermentation, which is also known as vegetable brining. So when we talk about this, the LAB helps preserve vegetables by producing the acid during the fermentation. So what happens here is, they have certain characteristic features under which this happens. The anaerobic conditions are required, and as a result of which the pH is going to decrease gradually, which results in improving the flavor and also preserving the food. So this is what basically happens in case of vegetable fermentation. Next we have the baking and sourdough. So LAB in baking, LAB basically work together with yeast to improve the sourdough bread quality. As a result, it improves the texture, enhancing the flavor and helps the dough in rising. Preservation. So preservation mechanism is nothing but how LAB protects the food. LAB creates condition that harmful microorganisms cannot tolerate, as a result of which we have acidification, bacto, bacteriocin production, and the competition with spoiling microorganism. So the important term is we have nisin, which act as a natural bacteriocin, which protects or which act as a target in protecting from the harmful pathogens that can cause the spoilage further. So probiotics and the health benefits. Some lab LAB bacteria improve the gut health and immunity, as a result of which we have better digestion, improved nutrient absorption, and also it supports the immune system. So as a summary and the key takeaways, LAB are beneficial fermentative bacteria. They're used in dairy, vegetable and baking. They basically preserve the food in natural form, and also it improves the human health as well as the digestion. So to conclude this, lactic acid bacteria are essential microorganism in food fermentation technology. Let's discuss about another new interesting topic in the upcoming lecture. Thank you.